Okay, so we're gonna start now. Hello, everyone. Welcome to this webinar, open day for the Genetics and Multiomics in Medicine Master's Program. We hope that today we're gonna answer all of your course-related questions and gonna give you more information about how it really feels to study on one of those master's courses. I am Daria Demieva. I will be chairing the webinar today. I am the alumna of the Genetics and Multiomics in Medicine Master's Program. I currently am a PhD student in the Center of Genetics and Genomics at the Renal Department of the Royal Free Hospital, which is a part of the UCL Division of Medicine. And today I will be joined by Dr. Horia Stănescu, the course lead of the Genetics and Multiomics in Medicine. And he will give you a presentation about the course, and followed by that will be a Q&A session where you can ask questions in the Q&A box on the bottom of your screen. So to present the speaker, Dr. Horia Stănescu has more than 20 years of experience in the biomedical research, with 4 years in the National Human Genome Research Institute in the USA. And after that, from 2007, he has joined University College London in the renal department. So he has experience he's currently a lecturer in the renal genetics and a primary PhD supervisor in the Faculty of Medical Sciences. So now, Dr. Horia, please, your presentation.
Thank you, Daria, for your kind introduction. Indeed, this program, I'm the lead of Genetics and Multiomics in Medicine, and this program officially started 3 years ago. But content-wise, we started long before, and it started with a series of informal research seminars of the Genetics and Genomics group within UCL's Division of Medicine, where Daria is also part of. She became recently part of that group. I will give you a short presentation and try to define some of the terms related to the course. And in the process, I hope that you will get a feeling about what and how and why we are doing things the way we do. And I will start with a quote by Walter Cannon. And this quote suggests that there is a profound understanding of the workings of the organism is a prerequisite to our ability to use medicine as a force for good. Only by understanding the wisdom of the body shall we attain that mastery of disease and pain which will enable us to relieve the burden of mankind. Now, the wisdom of the body is a poetic way to refer to homeostasis. Homeostasis is an important biological concept, and it was coined by Cannon. And it refers to the mechanism by which organisms in general, not only the human organism, maintain their stability, their equilibrium. And this quote implies that alterations of this equilibrium will lead to disease. In the context of medicine, we are driven by a quest to understand how diseases originate. And if we are to understand how they originate, we'll be better at diagnosing them and hopefully, eventually, better to treat them. So, pathogenesis, but genesis is also a Greek term that relates to heredity. And the science of heredity, genetics, is an interesting, refers to an interesting concept, which combines two apparently antagonistic notions, constants and variability. To give you an image to illustrate the beauty of variability of the living world, and this also underlines the need for constancy the intrinsic constancy and environmental constancy as well for its survival. So this is a coral reef. And the coral reef is a very complex system. In order to understand any complex system, like the reef or the human organism, one needs to analyze it and decompose it in simpler elements. This is the shape of one of the elements of the previous reef. This is the red coral. And the shape is suggestive for the way the tree of life is usually represented. Now, we are familiar with the tree of life. We usually don't talk about the coral of life, but it's still a similar metaphor which captures some salient points about the architecture of the network of life, its complexity, and the way information flows and branches out in order to generate beauty and ever-growing complexity. The material substrate of this complexity, of this information, is a class of molecules that are at the core of biology. They are called cemantides, molecules that carry sign, carry meaning, and they are at the core of our program. They are our objects of study. The relationship between these molecules, between cemantides, is known as the central dogma of molecular biology. Of course, calling it dogma is tongue-in-cheek because science is supposed to be anti-dogmatic. And we want to understand the world we live in by being flexible, not rigid, mentally and intellectually. But the one that coined the term central dogma had a wicked sense of humor, and I'm referring to Francis Crick. You see him here in a picture together with James Watson, and they are very well known for their discovery of the structure of DNA, together with Rosalind Franklin and Maurice Wilkins. Francis Crick presented this sketch of the central dogma at a seminar which took place at UCL. And also, he started his studies at UCL. He embarked on a PhD at UCL that was interrupted by the Second World War, and after the war, he continued his research in Cambridge. Now, the central dogma tells us about the interplay between cemantides, and this interplay ultimately will generate, will bring to light, if you want, the organism. And this bringing to light has a Greek concept. It's called phainein in ancient Greek. And this is the word that gave birth to the term phenotype. And phenotype means literally appearance of the individual. If we study the appearance of the individual, we study the phenome, which is the complete phenotypic representation of the species. And the science studying this is, the science of investigating the phenome is phenomics. Now, if the phenotype is brought to light, I thought I'll show you another beautiful picture by a master of light. And this is a Dutch painter of the 17th century, Rembrandt. And this painting is called The Anatomy Lesson of Dr. Nicholas Tulp. And in this picture, the human body is the main object of interest. Similarly, the human body is the main object of interest of the GMM program. Note how Dr. Tulp is using his hands in the process of dissection, of analyzing, of bringing to light, of enhancing the understanding by anatomy. So if you have in this image hands dissecting hands, if you want, which leads us to the next image of hands, drawing hands. And this is a drawing by another Dutch artist, Moritz Cornelius Escher. And this drawing suggests one of the aspects of why the human body is so fascinating, because the human body is at the same time the object and our instrument of research. So in our phenomics module, we are going to talk about instruments of research and instruments that allow us to investigate the phenotype. And obviously, they have expanded far since the days of early anatomists. What I'm showing you here is just an example on one of the first X-ray images depicting the bones of the hand of Mrs. Röntgen, which was the spouse of Konrad Röntgen. So you will study in the module of phenomics a lot about medical imaging and the way we use modern instruments to dissect the morphology of the human organism. But X-rays were also used to dive deeper in the invisible structure of the living organisms. This is famous picture 51, an X-ray diffraction picture taken by Rosalind Franklin in the laboratory of Maurice Wilkins. I mentioned them earlier in the context of the discovery of DNA, the discovery of the elucidation of the structure of DNA. And the elucidation of the structure allowed us 50 years later to read for the first time the entire genetic information encoded in the genome of a human organism. And this is the Human Genome Project. In the course of the genomics module of the program, we are going to extensively discuss the Human Genome Project, the way it came about, and the way it changed our approach to studying biology. And we now moved from the classical one gene after another approach, which was the classical genetics approach, to having access to the entire genome, roughly 20,000 genes, which can be studied in parallel, thanks to modern sequencing technology, which is going to be extensively discussed in the course of this module. Now, what does this mean for our quest to understand the pathogenesis of human disease? It means that we are using classical genetics. I'm showing you here a genealogical tree of one of the families we studied in our group to identify sequence variants that are linked to so-called simple or Mendelian disorders. But we are also using modern high-throughput genomic techniques to tackle complex disorders. And this is the output of such a study also performed in our group. And it's called a Manhattan plot. The colored stretches on the x-axis represent the chromosomes, and the signals on the y-axis are the regions where sequence variants are associated with one of the many causes of autoimmune disorders in this case. One of the most frequent genetic disorders is polycystic kidney disease. And what I'm showing you here is the way polycystic kidneys can be seen at different levels of magnification and using different imaging and phenotyping methods. But to connect the phenotype to the sequence variant, we need to understand how the transcription of the genes dysregulated in diseases appears. And this is what you see on the right side, wild type versus abnormal in RNA levels in patients versus controls. And this is the scope of the field of transcriptomics. Transcriptomics being the science of everything related to the second semontide, which is RNA. Moving from genomics to transcriptomics, we are stepping up to the next level of complexity. As I told you, in the case of the genome, we were dealing with an order of magnitude of tens of thousands, 20,000 genes. The transcriptome encompasses hundreds of thousands of RNA types. So this is the next level of magnitude. And if we want to push our understanding even further, we need to link the phenotype of the disease. And in this case, what you see here is a depiction of a neurological disorder that we studied. So we were able to link that phenotype to modifications in the structure of proteins. And proteins are the third semantide class. And the study of proteins is proteomics, which has a very ambitious goal. Ideally, we would like to characterize all the proteins in terms of shape, quantity, and their localization. And if we're talking complexity, proteomics is yet another step up the magnitude scale from hundreds of thousands of RNA molecules, types of molecules, to millions of types of protein molecules. And this increasing complexity needs to be addressed through a shift from classical biology, which relies on single molecule experiments and small scale modeling, to a systemic approach in which we model large data sets which are generated from different types of omics. To deal with the ever-increasing flow of information, we need powerful tools, and that will lead to automatization, and this is why computing science is very important. So we'll have heavy component of computing in our program, and this is shown here. This is a sketch of the program, which consists of 8 teaching modules and a research project. For the full-time program, the teaching modules are distributed in the first term 4 and the second term, the next 4. And the third term is dedicated to the research project, which will lead to the dissertation paper. In each of the teaching modules, the theoretical concepts are going to be covered in about 10 sessions or chapters. And one chapter usually consists of a lecture in the morning, followed by a lunch break, and then a practical session or seminar in the afternoon. The course has a strong emphasis, as I said, on computational methods. And this is why we have 2 modules, one of bioinformatics and one of advanced computational methods, that are the vertebral column, if you want, of the module, and will help you understand how to deal with large data sets that are generated through all the omics procedures. Our program wants to offer you a wide perspective of biology. We are focused mostly on concepts more than techniques, and we try to have an integrative approach. We are going to discuss not only about the science, but also how it interconnects with history and the history of science and how we got to understand what we understand. We are going to tackle some philosophical aspects, some ethical problems, some social problems, and we'll have interesting discussions, I hope. And as in this presentation already, you saw that I showed you some paintings, some drawings. We're also going to discuss the links to arts and to human culture in general. We are a very collaborative group, and we emphasize and encourage collaboration between lecturers and students. we say that it's not only you who are going to learn from you, but in the course of our discussions, hopefully we'll also learn from you and benefit from your questions and energy. And we also encourage collaboration among students, of course. You'll come from different backgrounds, and as science is a team sport, nobody can know everything. So we hope to foster an environment in which you'll also learn from your peers and start developing a network of knowledge. Of course, this collaboration is not going to be encouraged during the exams, but outside the exams, we hope that you are going to interact. We're also very interdisciplinary. We have lecturers that have a computational biology background. We have clinicians. We have researchers. We have theorists and experimentalists. And hopefully you'll benefit from the whole spectrum and learn about different ways of doing science. This is one of the beauties of being part of the research environment. There are many ways, there's a large freedom to tackle complicated problems, and all of your approaches are going to be welcome, and I'm pretty sure you'll find your way in this maze. Now, I wanted to show you a couple of pictures to give you an idea, a vague idea about how things might look. And here are some of my colleagues, the module leads and the lecturers that are going to be your guides in this exciting adventure that you'll be embarking on if you decide to join the program. And I'll end my presentation with a picture of your colleagues of this year. So this is a picture taken earlier this year. And this is the current group of students. And hopefully you can see from their smiles that they're enjoying interacting and learning and being part of this. Thank you very much for your attention, and I think we can now take some questions. Daria?
Yep. Thank you for the presentation. Very interesting. And the Q&A should now be open. So please send us all the questions you might have. Okay. So thank you for the first question. We have Susanna asking, will you cover epigenetics at all?
Yes, epigenetics will be covered in the first term. There is the genetics and epigenetics of disease module. And epigenetics being, well, the core of the regulation of genome functionality, it will obviously be tackled also in the genomics modules and the transcriptomics modules. And if it comes to, yes, so I think that's the short answer.
Yep, thank you. Okay, then we have an anonymous question. Would this course cater towards people who want to pursue a career in bioinformatics?
That's an important question, and we've had this discussion before with Daria.
Yeah.
I would say yes. Of course, 1 year is not enough to become a proficient programmer, but I think it is a very good starting point, and through the bioinformatics module, where you will learn to become power users of existing bioinformatical tools in the first term. And then in the second term, advanced computing will take you hopefully to the next level, where you will be able to even modify those bioinformatic tools. And some of you will be even able to design their own tools and create their own tools and improve onto the existing tools. So I think that this is going to be very helpful to set you on a pathway to becoming computational biologists. But it will take a lot of effort on your side and a lot of dedication and probably more time. Is this a correct answer, Daria? Because you are one of the students who I think were set on that path. Did this?
Yeah, and I mean during the course we also had discussions with other students about that, and I think it gives you a very good base to build upon, and the advanced computational module. When I told my friend who's a data scientist about the topics that we covered, he was quite surprised because it's quite advanced things about computers. Uh, and I think also if you really enjoy those modules, you can then take a research project in bioinformatics and in computational biology. And for that, you would also have some projects to show for employment or PhD in the future. So yeah, I think it's good if you want to pursue a career in bioinformatics. Okay, then we have a question from Sarvesh. Do you offer guidance or mentorship for students considering a PhD after completing the master's?
Well, I think this is probably a good intermediary step. This is a good stepping point to enter a PhD, to get to the next level, to start a PhD. Um, for instance, from this year's cohort, already 2 students were able to secure PhD positions even before finishing their master's program. So I think we're slowly getting even better at that. And I believe that through what we are doing and through our constant interaction and teaching and discussions, and we encourage you to engage us and to ask these questions. So I hope that this is preparing you indeed for if you consider this step to enter an academic career. Here again, probably Daria is best situated to tell you how this feels from the user's point of view.
Yeah, I mean, I am a PhD student now, and I have completed this master's program. And the Horia and the other people from the teaching staff were really helpful in like giving advice, finding the program, and with providing references as well. So yeah, I think there is guidance and mentorship for if you want to continue to do a PhD. Um, okay, another question from Wafi. Is GCSE, I don't know if it's O or 0 level English accepted for this program?
Um,
Mm,
I don't know exactly what level of GCSE English is requested, but this is, thank you, Emiliana. Emiliana kindly provided a link to the conditions for acceptance in this program, and you'll find that information, those details there.
yeah, just open the chat and the link should be available there. Okay, the next question is from Mariam. I'm sorry if I mispronounced it. What type of assessments does this program has, and what is the grading system?
There are, as I said, 8 teaching modules, so assessments vary widely, not wildly. So there's everything from written exams, multiple choice questions, short answer questions. Often there is the case that we have, there is the ongoing assessment and coursework and assessment of the participation and engagement of the students, but then it's usually followed by a written type of exam, MCQ or questions, and very often there's also a third aspect, which is an oral examination, which usually is in the form of either a short presentation in front of your colleagues, which should improve your ability to speak in public and to present your result, or graphical abstracts, or presenting a project. So there's a whole spectrum of evaluations. And the grading system is from 1 to 10 or percentage.
Thank you. Okay, another question from Vittorio. Are there any compulsory wet lab sessions or will it be solely computational?
There are also wet lab sessions. We're moving the way the program evolves. We're moving away a little bit from hands-on wet lab towards more observational wet lab sessions. That's also a result of our success in terms of number of students. Now the program has more students that we started with. So we were slowly moving from a smaller, more personal group, if you want, to something that is more institutional, which has advantages and disadvantages. But the point is, not everybody will fit in the lab. Well, we're splitting you in smaller groups, so you'll be in the lab and you'll observe some techniques, but there will be probably less hands-on wet lab experience than in the previous years. But we want you to be in the lab and to at least experience the generating of data, the way data is generated, that you'll be later working on and analyzing.
And I think, of course, if you want to pursue a research project doing a wet lab, that option is also there.
Yes, that's an excellent observation indeed. The projects, you can choose between data generating projects, which are going to be heavily wet lab. And there's again, there a whole spectrum from cells to tissues to model organisms, and moving towards data analysis and computational biology and population genetics, maybe evolutionary aspects that you can tackle in your project, research project. The way we're seeing it is, if we're doing things right, the first two terms, the teaching terms, should prepare you to tackle the third term, the complex problem that you'll set for you during the third term in your research project. And there you can choose between what you'd like most, wet lab or dry lab, or there are also some projects which combine both of them for the most ambitious amongst you.
There is another question from Yoon asking what kind of topics can students choose for their research projects, but I guess you have already answered that with a wide variety of that. Or do you have anything to add to the topics for research projects?
As I said, there are topics from all the modules. So there are topics relating to transcriptomics, where you might compare the expression of a particular gene or a particular set of genes under particular circumstances in diseased models versus normal models. There are now topics about spatial transcriptomics. There are proteomics-related topics, mass spectrometry in particular disorders. Diana Canati, the module lead for proteomics, has a particular interest in amyloidosis. So she usually offers a couple of projects relating amyloidosis, related to amyloidosis pathogenesis, and usually applying mass spec as a technology to tackle the data. As far as genetics and genomics are concerned, well, as I said, we have evolutionary projects. We can study ancient DNA and its relevance to the current phenotype of humans, and how sometimes we pay the price for previous adaptations, and some disorders can be manifestations of what earlier was intended to be adaptative in evolution in phylogenesis. So from studying ancient DNA to studying populations, differences between say populations in Southeast Asia and Northern Europe, and again with an emphasis on evolution and adaptation. GWAS related projects, that is genome-wide association studies, to identify loci that might be relevant in a huge spectrum of disorders, because don't forget, this is the division of medicine. So the PIs, the principal investigators who are offering those projects, are some of the almost 1,000, if I'm not mistaken, PIs from the Division of Medicine. And these are researchers and clinicians from all possible clinical specialties. You'll have neurologists, you'll have cardiologists, nephrologists, you name it. And they have experience with all kinds of research tools, again, from wet lab, from tissues, from cells, from CRISPR and Cas to the latest modeling techniques and statistics techniques that might be applied to data analysis.
Thank you. Another question from Susanna is, I saw that there is an option to do a certificate rather than the master's. Can one switch from one to the other? Say start with the certificate and then commit to the master's halfway, or downgrade from the master's to the certificate halfway?
I don't think we offer the certificate anymore. I'm sorry about that. I think this is, we need to update our description. Okay. Yeah.
Yep. Question from Wafi, is prior knowledge in bioinformatics or programming required?
Well, the short answer is no. We hope that we can take you from zero, and it's going to be a steep learning curve, but we are starting, as I'm telling you, with assuming almost nothing, and starting from the beginners level and users level, and then slowly working towards the understanding of the use of bioinformatic tools, and then only in the second term pushing the envelope a little bit harder and trying to move you towards some coding and elements of algorithms and understanding what's happening under the hood. Of course, having some previous understanding won't hurt, but again, we are really trying to start from zero and try to elucidate the basic concepts and basic approaches.
And I might add that we had students on my year of very different levels of knowledge. For example, with Python, from like absolutely zero beginners to people who have already used it in their work. And it was also a great opportunity for students to help each other out. And by the end of the course, we were all on a good, solid programming level. Anonymous question, what is the acceptance rate for this program?
Last year's acceptance rate was around 20%. We had a lot of candidates.
Okay, thank you. And another anonymous question, would the course touch on AI at any point?
Yes, this year there is a session particularly dedicated to machine learning and AI in the advanced computational module.
Thank you. Okay, another anonymous question. Can we get in touch with previous students to ask about their studies and preparation?
We already are in contact with a previous student, and yes, of course, you can get in touch with them.
Yeah, I think you can do it on LinkedIn. That would be the easiest option if you're.
I think here Daria is the best.
Or just I guess email me. Okay, question from Samran. What do you see graduate working as after finishing this program? Well,
That's a question that we asked ourselves when we started this program, and we hope, I mean, we know that multiomics and the genomics technologies are going to be more and more important in all fields of biomedicine. So from this point of view, what we hope is that we will have professionals helping in clinical medicine, and there is a move towards infusing the novel approaches of multiomics in the day-to-day life of, say, NHS, the health system in England, and I'm pretty sure that this is the case all over the globe. So health professionals will need to know about these new techniques, and you might be some of those health professionals working in clinical settings. Of course, we also hope that at least some of you, so far, I think it's been quite a high proportion, are going to embrace an academic career and are going to become researchers and hopefully help us. As I said, these are complex systems and complex questions that we're tackling, and there's still a lot of work to be done. So we need researchers, we need clever people, enthusiastic people, people with a passion for that. And I hope that, again, you will be those next gen researchers, and hopefully, if we're doing things right, we'll help you become those researchers. Why not? You can also start your own businesses or be part of existing businesses. Big pharma always has the need for data analysis, for computational biologists, for people who understand not only bioinformatics, but as I said, also the way this data is generated. So I think this is also going to be an advantage. You'll be confronted with all these omics techniques, and that hopefully will place you in a good position to hopefully secure places in big pharma. Or, as I said, start your own companies, startups. We are hoping that some of our alumni are going to be successful also in business and industry. We also have and had some students that come from different perspectives. There is at least someone who wants to understand biology from the legal perspective, wanting to inform their legal practice, and those are the ethical, legal, and societal aspects that are, of course, gaining from your understanding of the basic mechanisms of biology and the techniques that we're using to research biology. Am I missing something, Daria?
no, I think you covered everything. Thank you. And Emiliana added to the chat that the prospectus page mentions that the master's program is master's program only without the certificate. So that's the previous one. Another anonymous question: will we receive guidance or mentorship for the research and dissertation?
Yes, of course. So you will be assigned. Well, it's not that you will be assigned, actually. You will find the process is such that there is a list of potential projects and a list of students. And we encourage the students to not only go through the list, but engage with the potential PIs that are going to lead their project. And you end up contacting a few labs and in the process learn a little bit about what they're doing. And hopefully, in the end, everyone will find, well, not hopefully, surely, in the end, everybody will find a project that they will like and will be in an environment that is supposed to foster them, to help them. And the PI that offered that particular project is the one that is going to mentor them through the process. And this is still part of the learning experience. So you won't be just pushed into deep water and left to swim. There is, of course, going to be a degree of independence because we want this experience to be similar to a real life experience, and you will be in a real laboratory and you will interact with your colleagues, but these are going to be more experienced colleagues. These are going to be, as I said, PIs and PhD students and postdocs, and you're going to benefit from their input and feedback and advice. And I think that is pretty much the way it should be.
Thank you. A question from Wafi. Is this course suitable for someone interested in precision or personalized medicine?
Well, I would argue that it is indeed. In the Division of Medicine, there are now two programs that would be suited for that. It's the GMM and the Personalized Medicine program. There are obvious differences. We are possibly a little bit more geared towards the research aspects, and they're a little bit more oriented towards the practical clinical aspects, if you want. So there is some overlap. There are also at least 3 modules that we share. That is the genetics and epigenetics of disease, the bioinformatics module, and the ethics and multiomics module, but we also share some of the advanced computing and genomics resources. So there is some overlap. But the short answer is yes, I would say so.
Another question from Sarvesh. Are there any books or materials that prospective students can go through to be better prepared before the program begins?
Well, this being an ambitious program, basically Multiomics covers the entirety of biology. So I would be hard pressed to suggest a list of reading, because pretty much everything you read in the realm of biology will be useful. But if you really want to be prepared, probably a very good place to start with would be the Molecular Biology of the Cell by Alberts et al., which is now in its 7th or 8th edition, if I'm not mistaken, and which is an excellent starting point.
Thank you. And another question from Sarvesh. How many students are accepted to the program each year?
As I said, we have been quite successful, I'd say. We have a lot of applicants. This is why we increased the number of accepted students. We grew from a small group, a really small group. For a while we were around 10. Now we are hovering around 20, and I think we'll keep for now the number around 20.
Thank you. Anonymous question. What is the passing rate for this program?
All the students that started so far passed.
And is the course only based at the Royal Free Hospital campus?
No, the course is shared between the main campus and the Royal Free campus. I think in the first term it's more on the main campus, and in the second term it's mostly on the Royal Free campus.
Thank you. Okay, that's all of the questions answered for so far. If anyone has any other questions, please let us know in the chat. Otherwise, I think that has been very comprehensive session. Yes, I highly recommend this program myself. I really enjoyed it. There is a lot of opportunity for collaboration between students and with the staff. And okay, oh, we have more. We have Susanna. How many mature students do you get per year?
We had a couple of them each year, 2, 3.
Yeah. It's really, I think the program is open to anyone. So
Yes, we have different backgrounds. As I told you, we encourage people from different backgrounds. So it's not only from biosciences. We're encouraging computer scientists and mathematicians, statisticians, as I said, lawyers, business persons. So it is diverse from every point of view, geographically, from the background and formation. Also age-wise, there is a certain spectrum. So we have some mature students.
And do you have tips for writing a compelling personal statement for this program?
I would say be honest. This is the most important thing. So we had this discussion earlier. It's not supposed to be a marketing exercise. Ideally, we want to get that match in which you find a place that you fit in. And in order, this is a two-way process. In order for us to be able to tell you if you would fit and enjoy and thrive in this environment, we really rely on you being honest and telling us what you like, what you want, what you think you can do, and that is extremely important. Don't oversell yourself, because we are not expecting, how should I say, our reason to be is to teach you. So we're not expecting a high level of knowledge to begin with, but we are expecting the right attitude and, most importantly, the right motivation. We hope that you really want this. And if you don't want this, if you won't enjoy it, truly enjoy it, then this is going to be a failure and a loss of resources and a loss of time, both on your side as well as on our side.
You and a question from Wafi, is CRISPR going to be taught in this course?
Yes, this is going to be covered in the transcriptomics module.
Yes. Okay. We're out of questions again. And I think we're good to close it down and go for lunch now. So thanks everyone for coming, and thanks a lot for the questions. It's very great to see so many people interested in the course. And yeah, good luck with your applications. And thank you, Jorge and Emiliana.
Thank you very much. It's been a pleasure.